Hi, everyone. I'm excited that so many of you could join this webinar. I'm Lara Heij, a liver pathologist and scientist. I'm Dutch and I work in Germany in the University Hospital of Aachen. My science work is mostly focused on cholangiocarcinoma and the tumor microenvironment, and that's why I'm very happy and excited to introduce Dr. Vincenzo Cardinale, because he has been doing very interesting research in this topic and combining translational research into the digital AI tools. Before we go there, I would like to invite you all to switch your cameras on. It helps Dr. Vincenzo not to talk to the black hole. And also we invite you to make this an interactive session, so you are welcome to raise your hands or ask questions in the chat. We will keep an eye on the chat and try to interrupt whenever this is possible. Also, my co-moderator, Professor Bissan Al-Lazikani, will try to organize your questions. The last 5 minutes, this is also important, we will keep for the feedback survey. I know it's not our favorite topic usually, but it's important. I've seen how much work is put into the preparation of these meetings, and only if you give us feedback, don't be mean, just be honest, we can improve ourselves. Now, I'm very happy that Dr. Vincenzo Cardinale will take over. Most of us know him already. He doesn't really need an introduction. We know him from the ENSCCA, and now he crossed the ocean. He's in Boston, and also the other side of the ocean, we'll get to see him more. He now has a visiting faculty position at the Beth Israel Deaconess Medical Center. And of course, he still has his affiliation in Rome at the Sapienza University. We will talk shortly and introduce his work. And then he will also the introduction of cholangiocarcinoma and the challenges. And then he will dive into the biological and digital twin. For that, I think we have housekeeping disclaimers. We all approve and agreed upon recording. And please answer the survey. Vincenzo, the floor is yours.
Thank you, Lara. This is very kind of you. This introduction is really helpful for me. And of course, I want to thank the organizer, first of all, Cholangiocarcinoma Foundation, Stacy Linsdale, and in particular, Professor John Vallée, that at the end was the main reason this is happening today. But of course, I have to also to thanks my European friends, really friends, great friends. And I mean, today is 10 years, not today, but 10 years of European Network for Study of Cholangiocarcinoma, 2015 and 2025. I remember when we met me, Jesus Banales, Domenico Alvaro and Guido Carpino, TIAS 2015. So I think we should celebrate a little bit our achievements as international group of people devoted to patients and science. So my disclosure is relevant today because, you know, when we are speaking about new technology, you should not forget that you are a human. And for me, most importantly, I'm a physician. Let's say I'm a human physician. So I should always keep this in mind. Okay. So as already Lara presented, this will be like, I think more a debate. We should really, let's say, we can think that we should stop for a while and think that we have a new avenue in front of you, in front of us, and how we want to face this new time, how we want to embrace the challenge, especially not for us, but for the patients. So this is key. And this is why I think Joan invited me today to give this talk. And I think it's very relevant that is endorsed by a charity, the Cholangiocarcinoma Foundation. Okay, but okay, we are aware very much that Cholangiocarcinoma is just not a challenge as a burden. Is a big challenge as a burden, but behind and beyond is a biomedical challenge. We should not forget, and I'm very happy that today we have two pathologists here. They take care in each single case, each single patient about, let's say, outlier, very complex kind of information. Okay, so we should really be careful in the construction of every kind of new knowledge, to take in consideration that we should have a very multipolar, multidisciplinary, interdisciplinary point of view on complexity of the disease, and put, first of all, before the patients, okay? So the biomedical challenge, okay, of cholangiocarcinoma. Okay, the burden, unfortunately, we know that this burden is hitting more and more because the increasing incidence and still mortality. You know, Professor Vallée know very well that many of the big cancer, like colon, like breast, like lung, have at least something in prevention, like screening, and also nice approach in terms of precision medicine. But this is not the case for cholangiocarcinoma, okay? And perhaps this is derived also on our approach as a community. We should think about this, I think, okay? So it's interesting, dear friend, that in 2015, 10 years ago, when we started with a bunch of friends, we were a very balanced kind of group with basic scientists like Jesus Banales, Guido Carpino, me and Professor Alvaro that are physicians.
Hello.
Professor Alvaro is the real example of a physician. And when we started on our way, and Lara is very aware, and also the co-chair, when you start when you are looking at a cancer, on the cells of origin, on the tissue of origin. And you can imagine that we did so long journey. We did. It's 10 years that we are making it happening. Okay, because with this approach of registry that are imbricated, we are responding exactly to this question. Okay, where the cholangiocarcinoma rise in histology, what the molecular feature has, and what is the clinical presentation, radiological presentation. And we did a great journey together. We should be happy and we should celebrate. And I have to mention again, Jesus Banales was the racing force behind this. Okay, so this paper is published in 2025. You know, Lara, this paper is a very nice paper. It's a review. And say for the first time that the cells of origin of large bile duct intrahepatic cholangiocarcinoma and exhepatic cholangiocarcinoma, today we should call periral and distal cholangiocarcinoma, have as cells of origin peribiliary glands, okay? And perhaps the intrahepatic small as the small bile duct, the interlobular bile duct, intralobular bile duct, and canals of Herring, probably. Why I'm putting this journey like this? Because this was publication in Nature 2020. And you can see here, Lara, this is amazing. This is a figure done by Guido, and also, of course, by Jesus and a friend. Here, we put together all histological feature with the cells of origin, tissue of origin, clinical presentation, molecular presentation, and everything segregate all together, even with the mutation, okay? We are very aware about this. Think about small intrahepatic and EDH1 or FGR2, okay? So why I'm putting this on a line? Because in 2022, only 3 years ago, there was the ECDO 3.2, the fifth ECDO classification, World Health Organization. And it stated, and we should be very proud that the figure in this blue book was from Guido Carpi, okay? They stated that we should look at cholangiocarcinoma and classify histological in small intrahepatic cholangio, large intrahepatic cholangio, periileal and distal, okay? So this makes a perfect union of ECDO and ECD, okay? Keep this in mind. And ENSCCA, Guido, and all the friends contributed here, even Lara, demonstrate for the first time that the biomarker that has been created using this new classification and easy, very easy data like pulse positivity, just a simple immunohistochemistry, and clinical data like C99, liver test, and radiology help you in understanding biological behavior of cholangiocarcinoma. We should advocate to have this in the future clinical trial as a community, I think. This is, you know, I'm going crazy on this. I mean, I won't be very easygoing and calm, but I think we should advocate a little bit at least. I'm a gastroenterologist. I try to do translational research. There are pathologists today. But you see this beautiful paper by our friend Angela. Angela Lamarca is great and a great scientist together with Joan Vallée. I mean, look at this paper. It says that when you have liver propagation, liver infiltration within the liver parenchyma of intrahepatic cholangio, this means that you have metastasis. And Lara knows why. Lara knows why, because when a cancer passes from a tissue to another tissue, large bile duct to liver parenchyma, this is a metastasis, okay? And this paper demonstrates this, okay? I take this this slide by Jesus, and I thank him. I didn't change a lot. So, liver transplantation for intrahepatic cholangiocarcinoma. As a gastroenterology, I think I can say you, I think that the intrahepatic on liver cirrhosis will be great on liver transplantation and surgery. Perhaps will be not the same for the large intrahepatic because it's spread in a way that you cannot predict really well with a cross-sectional image. Okay, you cannot really understand very well. But until we don't include this classification in clinical trial of liver transplantation surgery, we will never know this, unfortunately. But it's also the same, dear friend oncologist, for the clinical trial in advanced patients. Is exactly the same. We have, unfortunately, a big impact of understudying, under-recognized complexity of cholangiocarcinoma in clinical trial. Because as we were discussing with Lara, we are not really, let's say, working a lot of outlier. This is not an issue now. I will face this later, but perhaps, you know, World Health Organization say we want to better classify cholangiocarcinoma because the name of a disease is everything. Lara can teach. We can have a beautiful lesson about what is nomenclature. There's a lot of Latin there. Nomenclature, classification is first starting. And there was, I mean, I have Guido Carpino and Eugenio Gaudio. They are anatomists. They know very well this. So here in this paper about pembrolizumab with Gen cheese, we are seeing a trend of a better response of intrahepatic. Think if we even classify in small and large, okay? Because thanks to this beautiful systemic review by Giovanni Frega and by Angela Lamarca, we know that perhaps intrahepatic has more PDL1 and microsatellite stability, okay? Perhaps the small. Lara, you can do this study with Guido. Let's do. Let's see if PDL1 in small versus large. And it's the same for when you add also anti-VEGF monoclonal antibody, perhaps you should expect the same. Why? Because the small intrahepatic is quite similar, at least in origin, with the hepatocellular carcinoma, where the standard of care is indeed anti-VEGF plus immunotherapy, okay? And it's also the same when we are looking on the other side of the story, the exhepatic. So there are some intrahepatic that clearly resemble the periilar, are the large one. So what about HER2 inhibition? We don't know. So let's go on real topic of today. And now, dear friend, I have to anticipate you that I'm putting some philosophy here and ethics, because we are facing a completely new field, new avenue. So, and I was doing so because this is experience. Doing this study in Italy, the digital twin of cholangiocarcinoma in the D3-4-Earth big European project that Professor Panebianco is leading in Italy. Every week we meet, and I was becoming crazy because what we can do here? I mean, how we want to construct the digital twin? I make many questions to me, and then I find the answer in philosophy. I have to tell you. So, because when we are speaking about digital twin, we are speaking about precision medicine, not more about how we prove the effectiveness of a drug, that is the randomized clinical trial, okay? Okay, but we want to create a new tool for the precision medicine. And when you have to consider tool for precision medicine, dear friend, you should consider the complexity. And the complexity is you can see here amazing cholangiocarcinoma. Because cholangiocarcinoma perhaps, and I want to know what you think, perhaps it's more heterogeneous cancer that we may have. But it's not because the cancer itself, but perhaps it's how we look at it. Because if you don't name him properly, we will just end a dark forest like Dante, you know? So very complex matter. So how I see the issue here? I see the issue that the patients and the cluster of our patients are shaped by multi-factor, okay? And these factors are not independent, okay? And this is why they segregate together. Things about EDH1 and things about also the segregation of EDH1 with small bile duct classification, okay? And dear friend, something that is important for people that want to have surveillance and screening, this complexity illuminate also this part of the story. So the preclinical, the preneoplastic, think about PSC, how we can depict the journey of patients if we use the same tool for everyone. We will never be there, okay? So paradox in medicine. I know perhaps I will even to not put the credit to me, because I think this is very obvious to everyone. So we are in a situation, historical moment, where we have a lot of technology. But how we use this technology? This is the question. How we use this technology? We use really to understand better the disease, or I don't know, perhaps we use the historical kind of criteria that are perhaps based on how I look at patient in my ambulatory and say this is diagnosis X or Y, and then we use just the molecular characterization at any level, any domain. I mean, by losing the interpolarity, the fidelity with the patients. So if you lose in this journey the fidelity in the pipeline, clinical work, clinical characterization, and molecular, then you are lost in the translation. This is how I see the issue today. And this is the ethics, the philosophy. Sant'Agostino say, we are what we love. We don't forget this. So we won't take care of patients. This is first things. And Dante say, we were not done to be like bad, like bruti, but to follow virtue and then knowledge. virtue, and knowledge. Okay, I think this is very relevant, and this brings me to consider that my patients is my scope, is my way, and my instrument. Okay, and so from this start my consideration about you know, how we want to consider our study. I think the first consideration is not what we are not dealing with objects, we are dealing with people. Is what is who. Responsibility means who we should respond to. Is the who, is the patient. Patients. Okay. So what is digital twin? Digital twin is something that is, let's say, old. It's an old concept. Okay, because digital twin was first created by NASA many years ago to trace, to replicate, to simulate the journey of a shuttle and see how in this journey the kind of decision that they take in this beautiful room then affect the destiny, the fate of this journey of the shuttle. Okay, so it's very easy concept. Is an adaptive, is an interactive data exchange between the reality and the virtual world of the data. But data are real. Okay, data are real. So depends how we consider the data as like capture, extraction, everything. This is quite important. It theoretically is very easy, practically very complex. At the end, we are using data-driven model like AI, you know, but also mechanistic model. I mean, it's not easy to understand how they can imbricate together. I was speaking a lot with Lorenzo Manganaro that is a scientist, a data scientist with a company, IOZone, that I will mention today. So we have hybrid model with data-driven models and mechanistic model. Mechanistic model is more hypothesis-driven. So you use your knowledge about the phenomenon, and then you are constructing a model. With the data, you are just driven by the data itself. You don't construct anything. When you put together this, you have hybrid model, okay? And this may help because mechanistic model work like a catalyzer and help you to have more readability and more understanding of the data-driven model, okay? So descending from philosophy is easy. Descending from ethics, the patient is the scope, digital twin is a tool as biological twin, okay? And this was created as digital twin, and concept digital twin is mainly for responding to value-based care. That is 5P guideline: preventive, precision, participative, personalized, and predictive medicine, okay? We should keep in mind that there are some rules, some guidelines to consider. So very relevantly, we should consider this that not me, but the National Academy of Science and Engineering and Medicine create a white book about digital twin and put the cancer patients as an example of digital twin in medicine, because cancer patient is the one that we are seeing more deeply and widely, taking all kind of information, all kind of data at different domain, and we are managing and following very closely. So we may give a lot of input in and out, a lot of input in this recirculation of data that is dynamically and is changing over time, okay, like our mind. So at the end, what do you want to do? You want to predict what is going on because this really happened. So when a cancer patient is progressing, it's too late. But with this tool, as Julian Calderaro, our friend, put in this new beautiful picture, you see, I cannot really put my screen, but you can see the in the upper side is our way today, and below you can see with artificial intelligence, perhaps we can scrutinize, we can have a mirror that may go deep in our patient without dissecting the real one, just the virtual one. And you may understand before what is going on in his body, in the blood, in the tissue, before that this patient come to you and say, oh, I am very sick, I cannot do anything, I have a fever, and so on. This is clinical presentation. But I am Italian. I come from South Italy. I'm very proud about this. And in Naples, we do very nice glove. No, I'm not able. This is a very artisanal way. It's a tailored kind of glove, beautiful. But the idea in virtual twin, digital twin, should be seen like this. We should create a glove on our patient, okay? Not thinking about chatbot that work in the way of generative AI. We are not still there, but we can come there, but only if we keep working of annotating data like Lara and Guido is doing with the hard work, like the registry is doing today, hard work again. And then we can go in the more foundational models and generative, but this is not the time, okay? Because as Dante say, we should follow the good way to go to the optimum. We should pass from the dark forest. We are not ready to go in paradise. We should pass from the dark forest of the hard work of the experience of every day. And you know what is the experience of every day for me? Is seeing a lot of patients, and I love it, okay? I hope Americans can enable me to do so soon. Okay, so sorry for this consideration. So we have a lot of challenge. I'm not the one that is stating this. Is in this white book, and here I have to acknowledge America. America is doing a great job with this white book, because we're saying that we have still philosophical challenge, we have ethical challenge, and most important challenge is trust between clinician, patients, and industry. Otherwise, we will be never there. I can tell you, without trust of our patients, what we can do? So this is why I don't want to be lost in translation, perhaps anymore, because I think it's very evident that we can be lost in translational medicine when you don't advance from preclinical to clinical study. But important, you know, here philosophy, epistemology is a branch of philosophy that think about what is the knowledge. But what is the knowledge for us, a scientist, let's say physician, or other stakeholder like patient or industry? What is the knowledge? And I think that the value of knowledge is different for each of us, but we should take, I think we can take in consideration all the value and try to be a little bit, clever, and consider that we can use digital twin to respond some question and understand the pathobiology, and then with this holistic way, go in the precision, and then prove that precision work fine in randomized clinical trial. This is my, very humble point of view. But without interdisciplinary and multi-channel, we will never be there. And this is again, ENSCCA, Angela, my friend Marco Casadio. This was a nice survey about multidisciplinary. I have to tell you that this is a cycle. In the middle of a circle of this, let's say, ethical alliance, there is the patient, and the circle is made by all of us, considering also patient, association, charity, scientists, industry, and the institution, okay? This is a virtual circle of the ethical alliance. But we should use multidisciplinary, as we are doing very well, and the interdisciplinary, because I learned this from Professor Alvaro, and philosophy also teach you that the limit of a discipline is the exactly moment and the exact situation when you advance, because you are aware that you have a limit and ask to your friend to close the gap together, okay? This is, I know this is again philosophy, but I like it, okay? I think it's relevant. We should be really aware that discipline means, and we should have like historically, you have a lot of students, young people in this like boutique of the knowledge that grow with you, and we should continue to do so with our beautiful network that we are raising. So Here, the concept become a little bit more sophisticated, okay? Lara, I think I have still 15 minutes, okay? It's true or not? No, 15 minutes or 10 minutes?
5, 10, yeah.
10, okay, it's fine. We are done. Okay. This it was inspired by Lara. I thank her very much. We met for more than 1 hour last week, and we agree about our vision. So Laura say me, Vincenzo, I was looking mostly to outlier. They are demanding for me, but this is not. I mean, outlier cannot be depicted by a clinical trial, so we should be very aware of this. But Lara can do, Guido can do, scientists should do. Okay, they look for outlier. And when you think about digital twin, it's like a chatbot, but it's not a chatbot. Work like layer. Let's start from more superficial one. This is how we can embrace this. And then synergy again. Synergy between randomized clinical trial, longitudinal study, and digital twin. And then it's funny this. I ask an help to artificial intelligence because I was thinking what I can ask to my digital twin. And we can ask many cool things. And this is something that you can ask again to a randomized clinical trial. But perhaps, as Professor Deshpande was very nice to explain this better than me in Salt Lake City, you know, this may help you to really understand the diversity and not to be just blinded by the appearance. This is what the question that we can ask, like clustering, like what is the really determinant, not how they are beautiful or old, but how they are inside, okay, with their disease. This is relevant. And this again, I'm sorry to say, still philosophy, because philosophy helps also in the implementation. So we are looking this our patients by just machine, just like data. No, I don't think so. I am a doctor, I am a physician, and when I work with my colleagues that are scientists, I say, hey guys, I'm sorry for you, but I have to tell you that this patient is not easy. It's not easy to understand.
Everyone, good morning. Welcome to
It's not easy to understand for you. Thank you, John. And this is the way of a physician is the critical relativist approach. It's still philosophy. The way of the machine is strong objectivism approach. So we should, again, please try to merge the approach. Never fight, never fight, never violence in any situation. I was also thinking about this today. So here what I talked during my work with my friend in Italy, and this meeting every week was, I have to tell you, was a mess. Because we were not really understanding what was the direction. But then we were understanding that we are creating a research platform and contemporaneously a digital twin development, a study. You cannot do a digital twin without a research platform with all kinds of data, omics, liquid biopsy, even spatial omics and clinical data, radiology and clinical management. You cannot do anything. You should rely on a research platform. You can do this only if you have a big group, multidisciplinary and interdisciplinary group that can close the gap. And sorry, because I had to change, my office was, my computer was crashed, and you can, sorry. So, and what you put in a digital twin is relevant. We should be clever. We should to investigate, and we should do acknowledge. We should know the literature, the last clinical trial, all the information in silico data. We should put all there, but with idea and hypothesis. Otherwise, you know. And also, hypothesis-driven research help because create a mechanistic model that help you. Okay. So one possibility with the dominant domain interdependency that you can use a digital twin for drug repurposing. You know that the EDGM1 have a transcriptomic feature that are similar to some cholangiocarcinoma. There are no EDH1 mutated like liver flux. Do you know? It's so. This is so. What do you want to do? We want to treat the patient based just on mutation or like transcriptomic, for example. And here, we are human. I don't think that a data scientist, an engineer can see a patient, see you look like a cirrhotic patient with an intrabody small bile duct cholangio. I can tell you, I can do. Professor Alvaro can do every day because we do every day. Science, me, 20 years, Professor Alvaro, 40. So, and we see in Italy, so many patients, 100 per week. So we are like a big machine learning tool. So I think that we should, in this journey of patient, to trace the journey, we should all together and dynamically speak each other every time. And we should make question that we like, that we think is important. We should share this question with patients and then construct like interrelated kind of data set that should respond to specific question. My question, dear friend, I want to predict, I want to prevent cholangiocarcinoma. Oncology wants to treat as best as can. And these are complementary synergical way. So let's move at the end. More about implementation. We should create new figure, new scientists like data, digital twin expert, because there are so many things to know about all the process from the patient to the digital twin. And along this journey that is dynamic, we should consider the rules, the regulation. We should consider the ethics. And at the end, the exploitation. But if you don't start well, you cannot never exploit. And you know that there are more than 10 regulations that you should be aware that you should implement in digital twin creation. I have to tell you, more than 10. And this is Europe. Europe perhaps is better. America still not so good. And more important, we cannot collaborate because we don't have common regulation. You know, it's a big issue also in randomized clinical trial. But for randomized clinical trial, we are good. But with this, we are not good at all. But believe me, everyone, and I'm speaking to IMMF, to Ellen Moreman, to Stacy Lindsay, to Cholangiocarcinoma Foundation. We should never accept that digital twin will become a black box. Never accept. Never accept. We should be very aware, always surveil to avoid this. Okay? Because digital twin is not a chatbot, it's a medical device. Keep this in mind. Okay? And still, again, the ethical alliance could help. Ethic first. No doubt. Ethic first. Otherwise, without trust of patient, we will never be there. Never. But we can exploit. Yeah, we can exploit. I was speaking with Lorenzo Manganaro. He said, my company is ethic. It creates like the patent. You can see the patent. You can read the patent every time. You can implement in-house easily a standard, like very grossly kind of platform. But you can ask to my company that I do a very cool, very friendly kind of platform, and you can sell. Okay, this is how it should work. And this is what we're doing in Italy with a big kind of project, European resilience plan by European Commission, D3 for health, D3 for ration. Professor Valeria Parenbianco is the lead with Professor Alvaro, and we created Cholangio-platform. I was discussing with my friend oncologist, Andrea Botticelli, and Monica Verrico, how we can call this Cholangio-platform. This is the oncologist. You see, you can recognize. We have everything, everything. And with a lot of help of pathologists, of anatomists like Guido Carpino, scientists, there are a lot of names. So we are interpolating liquid biopsy with specialomics and radiology, radiomics, and clinical data. Very cool. And we are also having primary cell lines of cholangiocarcinoma. This is very relevant because the alternative way is to use cells to ask the cells, this patient could respond to this large number of drugs, which is just another tool, the primary cell lines. And here, you know, we are doing with El-Bardisi, Professor El-Bardisi, a kind of connection of Cholangiocarcinoma Foundation and ENSCCA for primary cell line. And this project is very nice. So my take-home message today is, I mean, I don't have really a message. I want to share with you that we should start a debate. We should be, let's say, close each other. We should never fight. We should be happy about what we do. And then we can really go very smoothly and nicely in the direction that is the best for our patients. And now, celebration. Celebration of ENSCCA, first of all, 2050. Jesus Banales organized when he was still young, organized this beautiful meeting. And you see how Professor Alvaro is smiling. The day I am somewhere. And there is also the mayor of the city, as was, and then it came the ENSCCA after, and the first meeting with Helen, with Stacy, and these are the other photo. Our evolution, thanks to cost action that gives some money to do networking, first cost action European Cholangiocarcinoma Nation, and then the biliary tract precision network run by Rocio Machas and Chiara Bracconi, and they are doing a great job in continuity with the European Cholangiocarcinoma Network. We passed through COVID, everyone know, but we were out COVID very soon, and we were working as a family. I have my wife, yes, my beautiful wife, Melissa Gay, was the project manager, and we were all together, Jesus and my friend, now I have a lapsus, the beautiful guy that is up here at Organize Madeira, Rui Castro, a great guy, and we were a friend there, I can tell you, as a family there. And at the end, I have to apologize, especially with IMMF, because I have a problem. The first presentation was in my lap that crashed, but I have to acknowledge, first of all, two important people, Ellen Morement, Stacy Lenstey, IMMF, Cholangiocarcinoma Foundation, because for us it is everything. We are the tool, they are our scope, and we should really stay close to them, continuing to do our job as best we can. Thank you very much, and thanks to all my friends in this beautiful journey, okay? Thank you. I'm happy to take questions. I know in philosophy, you may be very wide questions, so it's fine. I put the presentation, okay? So I can on the
Thank you,
computer of my
Vincenzo.
friend Pinzu.
There must be questions, I think.
Thank you, everyone.
So,
Hi, Dr.
Yeah, go ahead, please.
Hi, Dr. Cardinale. It was such a nice presentation, and you put everything very well. And so I had a few questions related to radiomics, like kind of in the starting of your presentation, there was a one slide that I was kind of like I wanted to know a little bit, and I was just kind of, so I am Yashvir Singh. I am working with here at Mayo Clinic, more application of artificial intelligence and medical imaging in cholangiocarcinoma. I mean, like I saw that you are a physician. I don't know, it's just going to be like the more suitable question, but I was just kind of curious to know, like kind of what was the process that kind of like the one presentation I saw that, like the one slide I saw that, like where you are trying to just kind of combine, like kind of some pathological feature or something, like kind of the radiomics. So the
Yeah,
first,
I understand. I understand
yeah.
your question. Thank you.
So the one question was like kind of did you kind of do the radiomics thing, like that was multi-center, or like the and multi, like the different kind of vendor, like kind of the machine that like, or is just kind
Okay, I understand the question. So
Mhm.
because for time constraint, I want to answer. Thank you for the question. This is very relevant. This is an in-house work done
Okay.
by Guido Carpino with the histology registry. With I have the slide here. There are 6 European countries, 9 referral centers, 293 cases, multiple tissue biomarker, but no company. No company.
Okay, so the
No
question
company.
was like
So the radiomics were multi-center. It was
Mhm.
easy. You know better than me that radiomics works very well, okay, on CT scan. So it's very easily to standardize. This was done by Marco Rengo in Italy, and together with Guido Carpino, we are collecting CT scan data easily, and then put in correlation with histology classification, pulse positivity. Very easy, very easy. It's not because it's so complex. You can join. I mean, as I was discussing with Professor Vallée, I want to do something here. I contacted already this Professor Despande. I was contacting Nabil Erbardisi, and also you, dear colleague. Let's do something in US that is easily validating and replicating. But
Yeah.
likely there is no like patenting, there is nothing, so we can do together, okay?
So, and the another question was like kind of the, what was the integration strategy, like kind of the, how did you kind of like the integrate like the from pathology to radiomics? Because the integration, I have seen that kind of a major like the challenge that
Yeah. I think you are right. So if Guido Carpino is here, if I will be happy if we can allow him to
Okay.
speak, because he did the integration. You know, here is a very interdisciplinary and multidisciplinary, but you need like a leader, like a Guido in
Yeah.
this case, that was closing the gap, to take having the data by the clinician. And here we should really acknowledge the big work that the Lara Ischerardo and Jesus Banale did for the registry. And then Guido Carpino for the histology registry. And the radiology registry by was Analeo, it was Marco Rengo doing, and then the integration. But dear Ayash, I don't know your dear Yash, we can do
Yeah.
in Europe. I don't know if you can do this. I mean,
Yeah, I will
I
write
mean,
an
it's more
email.
complex, I think, in the US, I think, because in
Yeah.
Europe
Yeah.
we have a regulation that is the same for everyone. We work like, I mean, big. I want to ask Juan what he thinks, and also to Professor Al-Lazikani. I want to do this, something like a validation in US, but I think that in Europe we are working like more easily, because less constrained, because we meet many times together. We have this cost action that pay for our networking effort. So this is something of that is relevant for the future of us. We can discuss about this. So thank you, but
Yeah.
I think it's also nice to have other question. Okay, thank you very much. Thank you.
There's
Okay,
a
other
question
question?
in the chat. There's a question in the chat about how you might sort of account for or overcome temporal dynamism, especially that given the data are typically collected sort of as static points. So how would you represent dynamism?
Yeah, this was the main question that I didn't have many time to speak about my experience with the dynamically implementation of a digital twin, but this is what happened with us. We were facing a lot of issue because we were meeting, and it was like a big issue because there is a delay every time. When you are capturing data from clinical patients, this means clinical data, and always every time we're fitting with oncologists, please update the registry, and then please collect the sample, and then with the basic sign, please run the experiment. With the pathology, please rise the digitalization of slide. This is key, my friend. I don't know who did this question. Well, you understand very well what is the issue. And I mean, I was passing through. This is why I'm speaking of research platform. We cannot really do this with money. With money, I mean, we need a lot of money, but we need research platform. They need money, but first of all, we need the resource of human people, okay? Human people, professionalist, but that should be, let's say, humble to work together, okay? This is my answer. Okay, other question? Sorry if I want to have more questions, so please.
So while we're waiting for people, I've got maybe a couple of questions for you, Vincenzo, but if another question comes, we'll give it priority. So firstly, thank you for giving a shout out to the National Academy's report. We spent a very long time putting that together, so that was very good. But one of the key challenges, and I think it's more of a challenge in an area like cholangio, is the interplay between biological representatives and biological models. So there is a paucity of organoids, appropriate experimental models, because a digital twin works best when you can use it to make a hypothesis, you test the hypothesis, and you sort of go back and feed the digital twin to improve it. So what is sort of your comment on this platform would handle the way this interaction happens between digital and biological twin?
Yeah, Professor, this is a key question. I don't have an answer still, because, you know, the main issue, I think we are facing, one of the main issues, that is after the consideration of ethics and about how we want to really think about this tool, is how we implement exactly all the tools that are inside the big tool. So I'm thinking something like this. You know that we have a cancer vaccine today. We have even the idea of cancer interception. Cancer interception and cancer vaccine may work in two different ways. You may have just a tailored with a precision medical approach on the genetics, transcriptomic of patients, and then you have a lot of kind of modeling to understand the new epitopes and the antigen that you need to recreate. Okay, so this is like more, I think it's more like a mechanistic model, I think. But for example, for when you are doing cancer interception, you are doing not for single patients, because you don't have really the characterization, because the cancer is not there. It thinks about the Lynch syndrome. You are collecting information for a lot of patients. This is more about data. So I was even speaking with this company in Italy, because I want to do like a work like cancer interception in primary sclerosing cholangitis, for example. I was meeting this guy. I said, "Whoa, you have Lynch syndrome, but I have PSC. I want to share the knowledge all together. Let's do. Let's teach. We can do like a nice Joan, I don't know, and also my European friend. Let's do a meeting about cancer vaccine and cancer interception also with pharma. I think it's a good idea. This is an example. This is an example of digital twin. Okay. Thank you, Professor, for asking.
Well, while we're waiting, I just wanted to also say how important it is that you brought in the ethics right at the beginning, because something that we often ignore, sort of speaking as a computational geek, it's very easy to get excited about developing the technology and not thinking about actually what is the outcome that the patient is trying to achieve, not just, you know, the cure, but as we develop digital twins of individual humans, how do we anticipate that the patient is going to be interacting with the digital twin and how it can be optimized for the outcome that the patient requires? So I wonder what work you've done in this area, please.
So this is an wow, your question. I see your expertise in this question. You know, I'm not prepared to really answer to this. But what I can tell you, this is why we should stop, think about, you know, philosophy is important because what is knowledge for us? What is knowledge for the patient? And also philosophy is important because we may understand what we may answering with a digital twin and what we are answering today with the clinical trial. So I told you before that I think that the synergy is easy to understand because if we really work fine considering what are the answer to our question, we will put the nice response in a digital twin. This is responsibility. And then I think, as Lara was suggesting, we are addressing more the problem of outlier with digital twin, and then we will teach randomized clinical trial to make more precise the design. And here, dear friend oncologist, keep this in mind, please, because we need to be close together. I think I'm considered a little bit, let's say, out of a box sometime, but this is time to take this in consideration. I'm very glad that I mean, to Joan and to you, Chase, that you may have a common vision about this, because if I'm only me, it's not really well. But if you agree that this is relevant and you are important person, I think I'm stronger. Okay.
Vincenzo, can you please share the last slide of your presentation with the code for the feedback survey?
Oh, sorry. I was too, sorry, but we have 3 minutes, so
yeah,
we should do this beautiful survey, please. All of you should do. I will try also to do myself. So sorry, Joan, for this. We will solve the issue. Okay.
Is there maybe one more final question? Then I already want to thank you, Vincenzo, for your great talk. I'm looking forward to the future and also collaborating with you, and hope we can move forward.
Yeah, I think, thank you, Lara. Thank you, Professor Al-Lazikani. Please be all together, Europe and US. We need help each other. Otherwise, we will really not get there, because we are very complementary, synergic. I think we should really think about this. And Cholangiocarcinoma Foundation, MMF, and Cost Action, ENSCCA should advocate for, let's say, ethical alliance that will be really international, okay? Because such an expertise is so hard to find, okay? Thank you so much, everyone. I'm very thankful to all of you, and you, Chairs, for the nice moderation. Thank you so much.
Thank you.